Circulating tumor cells are cells which are delivered into the circulation from a tumor or from a metastasis. And the first description of the presence of tumor cells in the peripheral blood dates the 19th century. Probably a fraction of these cells comes from the primary tumor, and this can be considered as the very first step of the metastatic cascade, while another fraction may derive from the metastasis. That detection has been considered very important in oncology, and I would say that in the last decades many efforts have been made to develop a reliable method to detect the CTC. And this was not so easy because well, CTC are a very rare event. For example, in metastatic patients, we can find one CTC out of 1 million, 10 million of mononuclear cells. And another problem is that they do not have specific marker, but usually they share the same markers as normal epithelial cells. Well, first of all, to overcome the problem, as they are a rare event, more or less all the methods have a first very preliminary step of enrichment of the blood. And then I would divide the technologies used into two big groups. The first one based on nucleic acid. RT-PCR in particular is used to detect mRNA of genes. But as I said, these are not specific for CTC. So in this method, we must be very careful to search, to look for a good threshold to discriminate between normal or not normal value. Probably these genes are overexpressed in CTCs. And with this method, we are not able to count the cells. We just can say positive or negative, but we are not able to count them. Another approach is to use an immunological approach. So these cells are labeled with antibodies. And then, for example, using a cell sorter, we can count them. And very recently, a new method, standardized method, have been introduced. And this method allowed us to count the cells and to isolate these cells. For example, we use such a method, which is based on the detection of CTC, which is cells with a nucleus expressing cytokeratin but lacking CD45 expression. Yes, there are some trials ongoing, and we also had some trials. The very first we undertook was in patients with advanced breast cancer. And this was because we wanted to, as a first approach, we wanted to confirm and validate the results of our American colleagues who published the very first study in 2004, and they saw that the finding of 5 CTC per 7.5 ml of whole blood was predictive, was an independent predictor of poor prognosis, as in terms of progression-free survival and overall survival. And in the same study, they observed that patients in whom they found high level of CTC, but in these patients they found low levels after 1 month of chemotherapy, while these patients had a better outcome. So we enrolled in that preliminary study 80 patients, and in fact we could confirm that 5 CTC could discriminate between 2 groups of patients with different prognosis. And we also observed that looking at CTC during the follow-up, when we saw an increase, when we observed an increase of CTC during the follow-up, this was predictive of progressive disease at the following re-evaluation, clinical re-evaluation. So starting from this experience, we decided to move to other malignancies such as colon and prostate cancer, and also to see whether these CTCs are useful also in earlier stages of disease. I must say that in fact for colon cancer and prostate cancer, we now have some data saying that, in fact, 5 CTC for prostate, for advanced prostate cancer, and 3 CTC for advanced colon cancer are predictive of poor outcome. Well, I would say that apart from the prognostic role of CTC in patients with advanced cancer, as we saw, they probably have a role in detecting disease recurrence after the end of treatment. And another field of research is the characterization of CTC, and this is a very interesting and challenging field of investigation, of active research today. We are also involved, as other groups in this field, and, for example, we would like to better understand the characteristics of CTC and to see, for example, if cells found in patients with advanced cancer are comparable or not with the primary tumor in terms of genetic alterations and protein expressions. And another area of study could be to see whether therapy-induced effects on tumor tissue can be detectable also in CTC. For example, induction of apoptosis could be one of the possible field of research. So I think that apart from counting them, and it's important to say that now we have reliable methods to count them and to compare different samples in the same patients or tests performed in different labs, I think that the future will tell us that CTC are important also in other malignancies, and the characterization, as I said a few minutes ago, will tell us some new biological findings in the CTC and maybe will allow us to better treat our patients.